Ok, então diz-me quando clicares e eu dou 2, 3 segundos.
Ok. Agora.
Hi everyone, thank you for joining us for another session of the CNT meetings, now online. Thank you for your presence. Today we will learn evolutionary lessons from the Drosophila melanogaster for colonization. We will learn about how do history, selection, and effective population size shape evolution. And for that, we will count with the expertise and with the talk by Marta Santos, researcher at C3C, who is the invited speaker of today's CNC meeting, and to whom I thank having accepted this invitation for this CNC meeting. And before I give the floor to Professor Margarida Matos, who is also here with us and who will present researcher Marta Santos, I will also invite you for during the session, you can ask your questions or leave your comments about this presentation on the YouTube live chat that you have on your right here on your screen. So without further delay, I will now give the floor to Professor Margarida Matos to present our invited speaker. Thank you very much. Could you please turn on your microphone? Yes, thank you.
So hello everybody. It's a pleasure to introduce Marta Santos. She has been for a long time now doing her learning in evolutionary biology and then as a researcher. So she started with me, if I'm not mistaken, in 2007 or even 2006 as a volunteer in my laboratory. And she liked it because then she did her master with me. And it was really a very interesting master thesis that she did. And it was already focusing on the effective population size in response to adaptation to a novel environment. And after that, I think that you didn't regret the master because you wanted to do more, and she was completely crazy doing a PhD project in University of California, Irvine, where she did a mega analysis, a much bigger study that it was unique in the world. She will speak about it today. After that, she came back to Portugal, and she had several different moments of learning in SEDOC, for instance, and then she came back to our team, and so it's a pleasure that she is already doing her postdoc research with us in our laboratory. So I won't take any more time. So I'm sure you'll like the story that she's going to tell you about this long journey in her PhD huge experiment.
Thank you, Professor Margarida, for such a generous comment. And thank you for having me here. I'm going to start sharing my screen. Okay. There you are. Good? Okay. So good morning, everybody. Thank you so much for having me here. And let me start this talk with a little disclaimer that my talk is not slightly as cool as the one given last week by Luis, but please bear with me. I thought I could take this opportunity to tell you the story that led up to my doctoral thesis. It was a very long and bumpy ride, but also really cool, very fruitful, and extremely rewarding. I will now present my doctoral experiments on effective population size, which were fundamentally experimental evolution wrapped in a colonization scenario. It all started here at CETRG, at the time CBA, in Guida Matos lab, when I was completing my master experiments on the effect of small population size during evolutionary domestication of Drosophila subobscura. Using experimental evolution, I compared the evolutionary trajectories of life history traits of population of contrasting census sizes, with a large population with about 1,200 flies and the small population with about 50. The results I got matched the theoretical expectation. Smaller populations had both lower evolutionary rates as well as greater population heterogeneity. And although rigorous, these experiments were very low in replication, which is a feature of utmost importance in experimental evolution. So we wanted to do more and bigger experiments to address several evolutionary questions. And that led to my personal migration to Irvine, where I met my new scientific family. I bought a Porsche, and I survived using the imperial system, and I still don't know how to use Fahrenheit. I then joined the Rose Muller lab at UCI, where the best scientific conditions were met. I had this huge lab, highly replicated and outbred stocks of Drosophila melanogaster, with hundreds of generations of adaptation to the lab, and a very well-known evolutionary history. So consider the following scenario. After a forest fire or hurricane, a population migrates to a new environment where it encounters qualitatively novel nutrition and possibly including periods in which it starves. Its effective population size may be reduced in the course of migration or as a result of initial lack of adaptation to the new environment. So the first question that rises is how will the colonizing population evolve in response to such new environment? It is known that natural selection is able to produce rapid adaptive responses to sustained environmental change under propitious conditions like intense selection, abundant genetic variation, and large population sizes. A great example of quick adaptation in nature is the Drosophila subobscura colonization of the Americas, which later saw the establishment of of a latitudinal cline similar to the European one. So understanding the several factors that constrain evolution is one of the long-standing questions in evolutionary biology, and it was the focus of my experiments. First of all, effective population size is a major factor affecting the evolutionary dynamics of fitness and its components. Whether in the wild or in the captive environment, the evolving populations are very vulnerable to several genetic deterioration factors, which can reduce fitness and are dependent on effective size. Inbreeding, for a start, is known to decrease reproduction and survival of natural populations that normally outbreed, and the decline in fitness associated with inbreeding is known as inbreeding depression. Another factor that can lead to the population's extinction is the accumulation of mutations, deleterious mutations that can cause what is called a mutational meltdown. And also the loss of genetic diversity, which is crucial, as it limits the response to selection and decreases the population evolutionary potential, compromising its ability to deal with environmental changes. A classic equation predicting the rate of this loss as a function of population size is the loss of heterozygosity through time. On the one hand, since the middle term of the equation is a negative exponential, closed populations under drift are expected to lose heterozygosity exponentially with time, at a greater rates the smaller populations. On the other hand, and according to the Wright's fixation index, population with the same origin under strong drift will tend to fixate different alleles, which will increase the population heterogeneity. Each one of these genetic deterioration factors depends on size in a certain way. Inbreeding depression, loss of genetic diversity, and mutation accumulation are expected to be worse in smaller populations, and genetic adaptation or evolutionary responses are expected to be greater in larger populations. A population's performance and fate will then depend on the establishment of this equilibrium between genetic drift and natural selection, being the first more severe in small sizes, and the latter more prominent in larger sizes. So in the first part of my talk, I will approach this subject in bigger detail. That being said, interpopulation hybridization has been offered as a strategy to alleviate the negative effect of small population sizes, which can increase their fitness and decrease their extinction risk. Hybridization among small population may allow the recovery of some lost genetic variation, increasing their evolutionary potential. And the effect of hybridization on the response to selection under different population sizes will be more thoroughly presented in the second part of my talk. Many of the factors that impinge on successful colonization can also play a role on the evolutionary success of reverse colonization, which is when former colonizers return to their ancestral environment. Reverse evolution is by definition the reacquisition by derived populations of the same character states as those of the ancestor population. And it is known not to be universal, but contingent to factors like the time exposed to the novel environments, the differentiation from the ancestor, and the genetic variation of the character. Through reverse experimental evolution, we can study the factors that shape reverse colonization, which is a scenario of major interest within conservation biology. So this figure here shows a 50-generation long reversion experiment led by Henrique Teutonio, which is my scientific big brother, in the Rose Lab. And the results show that reverse evolution occurred, although it was contingent to the stocks that were used and the traits that were analyzed. So until now, these studies only covered population sizes that were big. And the experiments I describe here in part 3 of my talk come and fill in the gap with data on small populations. Another constraint on colonization is the change in the age distribution of the colonizers. It is a general evolutionary expectation that adaptation to novel environments shows strong age specificity. So this idea comes from Hamilton's theories, forces of natural selection, where the forces are very high at early ages, start declining with adult age until they reach an asymptotic plateau. The Hamiltonian force of selection theory is therefore relevant for the problem of the population, which is transferred to a qualitatively novel environment, like it is discussed on part 4. So this body of data obtained during my stay at UCI was compiled in my PhD thesis that was made of 7 chapters. The first presented the state of the art, then it was followed by 5 experimental chapters, and it was wrapped up in a general discussion. So before I proceed, I would like to thank all my devoted students with whom I worked for several years and who have taught me so much. These are just 21 of the experimental leaders out of 200 students with whom those crazy experiments would never, ever be possible. So with these 5 experimental chapters, 10 scientific questions were addressed, and I will revisit them a little further. The history starts in 1975, when Philip Truman Ives founded the original IV population from a wild population in Amsterdam, Massachusetts. So in 1976, Michael Rose brought those flies to the lab, and in 1980 created 5 replicates that were kept separate from then on. With the original IV called B0, we have 6 BI populations. 30 years later, we derived the experimental populations for my project. From each of the BI flies gave origin to 10 small populations kept at a census size of 50 and 4 large populations that were kept at the census size of 1,000. Half of which were subjected to starvation-resistant selection, and the other half were kept as controls, with a total of 84 populations. So the starvation-resistant protocol was pretty straightforward, and in the large populations, the flies were reared in vials for 2 weeks, after which about 5,000 imagos were collected and given 3 days of high-yeast diet. They were then subjected to starvation selection by giving them agar only and no food. The flies were checked every 4 hours, like having a small baby, and selection was maintained until 80% of the flies died. So the last 1,000 flies were recovered. And actually, they were not 1,000. There were just 500 females because all males would die in the course of selection. So those surviving 20% flies were recovered and fed, and their eggs were collected and found the following generation. And this were done for 25 generations. In the small populations, it was very similar. The populations underwent a similar procedure, started with 250 imagos and went all the way down to 50 flies, 25 inseminated females. During the selection protocol, the flies were getting gradually more starvation resistant, which caused a progressive increase in the life cycle length, which has consequences, and we'll see later. And matched controls were kept throughout the experiment. So every few generations, independent samples of these 84 populations were assayed for both starvation resistance and early fecundity. I won't bore you with the details, but I just want to highlight that these assays were done very, very close to the maintenance environment and as close as possible, which is something that is very important in experimental evolution. So the set of experiments reported in this part of my thesis was the central study, and responded to the first 5 questions throughout the largest and highest replication experiment ever done in Mendelian populations. So I wanted to know how populations respond to novel demographic conditions, how life history evolved in response to those new harsh conditions, how will the effective population size affect the evolutionary response, if populations from the same ancestral source evolved the same way, and what are the relative roles of history, chance, and selection in shaping the population's evolution. And the main results that we obtained were the following. First, on the effects of population size on the direct response to directional selection. We have plotted here the starvation resistance of females throughout the generations, the large lines on the left and the small lines on the right. The control corrected data is here shown as the difference of each line with the matched control. And this was done to account for the significant changes in starvation resistance that were associated with the postborn reproduction. So in terms of direct response to selection, these populations corroborated the theoretical predictions. There was a clear temporal increase in starvation resistance as a response to strong selection, and a significantly reduced response in the smaller lines. In terms of indirect response to selection, we have plotted the absolute fecundity values for the beginning of the experiment and after 22 generations of selection. So the first case, which is in the absence of strong directional selection, fecundity showed the expected temporal decline due to the postponed reproduction. And that was significantly sharper in small populations, showing the clear loss of function caused by small size. Under strong directional selection, the results were less clear. The smaller lines showed a steep decline due to the combination of inbreeding and starvation resistance fecundity trade-off, but the large lines, despite the absolute drop, there were quite inconsistent trends across replicates. And that may be due to a relative lack of replication. Now, in terms of population differentiation, variation among the 6B populations is expected to occur due to the secular drift among them. So we expect a temporal increase in differentiation in the derived lines, and the stronger the drift, the greater the expected divergence. Here we have plotted the variance of starvation resistance standardized by the mean. And we tested the effect of size and the effect of selection. So we found 3 interesting things. First, higher variance among smaller populations when compared with their matched controls. And then a reduction of variance among small lines due to selection, and that matches the classic expectation, as higher fitness alleles increase in frequency and the least adapted are counter-selected. And lastly, we saw an increase in variance among the large lines due to the combination of uniform selection and drift, which may result in the fixation of different alleles, causing higher divergence than by drift itself. Later, in the course of their PhDs, Mark Phillips sequenced these small lines and their respective controls after about 50 generations of forward selection. And Jimmy Kizos, another scientific sibling of mine, sequenced another pair of starvation selected lines. These lines I created also while I was at UCI, and they were kept in large sizes. And this experiment was done 75 generations after. So in terms of mean heterozygosity, the large SEO populations showed a steep reduction in genomic variation, most likely associated with the starvation selection. And they saw higher interpopulation differentiation in the selected lines. In the small lines, no significant differences in mean heterozygosity were found. And it seems like selection did not trigger great reduction in variation. And in terms of population differentiation, there were relatively high levels of FSD, which were similar in both lines. So the reduction in size seemed not to produce extreme reductions in genomic variation, but caused high levels of population differentiation. And here we have the CMH test in both experiments with the threshold marked in red. And in terms of SNP differentiation, the SEO analysis revealed a large number of SNPs across the genome that change in frequency, but no significant sites were found in smaller lines. So this increased genetic drift due to size seems to highly impair the identification of candidate SNPs, despite the high replication of the experiment and the clear phenotypic response to these lines. So to wrap up, population size, it is really, really important and is something that we need to maximize in experimental evolution. So next, in an attempt to respond to how hybridization at different sizes affect evolutionary dynamics of populations, we did the next set of experiments. Starting from these experimental populations and after 15 generations of forward selection, all replications within each BI selection and size regime were crossed in a massive hybridization scenario. And this doubled the number of experimental populations, and the starvation-resistant protocol was resumed. So our expectations were the temporal increase in starvation resistance and anti-parallel decline in fecundity post-hybridization, and the smaller the populations, the stronger the effect in the evolutionary rates. Here we have the control-corrected data for starvation resistance shortly after hybridization at generation 16, and then 10 generations after hybridization. What we saw was a general improvement of starvation resistance and worsening of fecundity in all lines, but no significant effects of hybridization. So maybe the loss of genetic variation could have been consistent across lines, making them very similar and thus insensitive to hybridization. Or perhaps small populations lost environmental homeostasis due to inbreeding, increasing population environmental variance. And this effect might have led to higher interpopulation variance without necessarily increasing additive variance after hybridization. And another hypothesis depends on the genomic foundations of life history traits, that evolution led to smaller genome-wide variation loss than classically expected, so no significant effect of hybridization would be expected. Prior research from the Rose Lab has shown that relaxing selection on starvation resistance leads to a clear evolutionary reversion of the character, but in large populations. Here we're asking if such clear response also happens in small populations. And for that, we picked up the small size populations that had undergone selection for 15 generations in their matched controls, and we returned them to their previous long-standing selection regime, the ancestral environment, which had early reproduction, ad libitum feeding, and no food supplementation. And we did that for 21 generations. So with the creation of these 16 new populations, we expanded the experiment to 228. So first we checked if the populations were differentiated from the ancestor, the bee flies. Then we tested if they were similar to the bees after selection. And if so, then reverse evolution occurred. Here we have the trait value in the y-axis tested after 5 generations and then after 21 generations of reverse selection. There's the ancestor bee on the left and the population, the experimental population on the right. So here we saw that the male starvation resistance of the RCPV lines matched the ones of the bee flies. So after 21 generation, there was no difference to the controls, showing a rapid and complete reversion of the male starvation resistance. In terms of the starvation resistance of the RSPB lines, we saw that they were initially different from the bees, and although the reversion occurred, there was no full conversion to the ancestor. And finally, here we have the fecundity of the RSPB. Flies that were initially differentiated, and after those 21 generations, it did not only equal the values of the ancestor, but it surpassed it. So it seems that reverse evolution to the bee ancestor was not impeded by the small size, by those sustained bottleneck, but had some contingencies on character and the evolutionary history of the populations. So the last part of my talk refers to the first experimental test ever done of the Hamiltonian wave. It was a very crude test, but still the first. So to check the impact of novel diet during a colonization event, we tested the age specificity, the age-specific mortality of flies that were fed banana, that is the control, orange, and avocado. And our results show that, not surprisingly, due to the lack of adaptation to those environments, small changes in diet can have very strong effects on longevity and age-specific mortality. Second, and following the Hamiltonian reasoning, a change of diet should have the stronger effect the earlier the diet switch occurred. So we performed a very crude banana-orange diet switch, where we switched the flies' diets every week for 4 weeks and checked their longevity. And the results did not corroborate the expectation from Hamiltonian wave, but indicated that the length of exposure to that novel diet, that novel nutrient, was the main factor that increased their mortality. And to further investigate this Hamiltonian age specificity of diets adaptation, we took advantage of Larry Muller, another PI from UCI, the UX and AUC flies, a 5-fold replicated stock of Drosophila that were exposed to urea early in age for hundreds of generations, and their matched controls, the AUCs, that were never exposed to urea. So we tested them, all the flies in both environments, and our expectation was of the loss of differentiation of mortality with age in the new environments. But the results that we got showed that the exposure to urea increased the mean longevity of the flies, regardless of the evolutionary history of the population. So no age-specific decrease in differentiation between urea adapted and unadapted population was found. But this doesn't mean that this hypothesis is not verified here for other life history traits like age-specific fecundity, and that was done later. And they actually saw age-specific fecundity confirming the hypothesis. So the last question that we had concerned another possible scenario of reverse colonization, where large populations long selected to novel stressful environments return to their ancestral nutritional environment. So in this scenario, and if costs of adaptation are involved, we expect that urea adaptation would lead to reduced fitness in the banana environment, particularly early in life. And conversely, at older ages, the performance may improve when it's returned to the ancestral environment. And what we saw was that the urea adapted flies had reduced mortality rates when compared to the banana adapted controls, providing no support, once again, for Hamilton's expectations. And this not preclude, again, the possibility that other life history characters may behave in accordance to it. And in fact, some ongoing research in the Rose Lab using other stocks and using other diets, a more ancient diet than the one that they have in the lab, showed age specificity of dietary adaptation. So to wrap up, reduced small population size caused significantly impaired responses to selection and stable candidate identification, increased the population differentiation, and shaped the signatures of history and chance. Hybridization can have very unpredictable effects on a population's subsequent evolution, and most likely won't rescue the endangered ones, and this is very important for conservation management. Evolutionary history is very important in a population subsequent evolution, and namely in a reverse colonization scenario. And small changes in diet can have significant effects on the age-specific mortality and pose a big challenge when a population is colonizing that new nutritional environment. So I'd like to thank my funding agencies, UCI, CEDRICHE, and FCT, my advisors, my scientific family, my kin family, my husband, but most of all, I'd like to thank my little boy, Zé Pedro, who showed me that there are things that are way, way harder than a PhD in experimental evolution. So thank you all for listening.
Thank you. Thank you very much, Marta, for your very interesting presentation and for letting us learn with you these evolutionary lessons, and looking forward also to the new lines of research that are coming from here. We now invite everyone that is following this stream and this presentation to leave your comments and leave your questions on the YouTube live chat that you have on your right here on your screen. While we wait for questions from you that you're following here this session, I also ask Professor Margarida Matos, who is also here with us, who has this very active also contribution also in a part of this research and follows very closely this research. If you have any comment, any question, also to Marta.
Well, a comment for those that are not used to maintain laboratory populations into experimental evolution. You may have the idea that it's very easy to maintain Drosophila. Well, Drosophila is easy to maintain in those like inbred isogenic stocks. Even so, it's time-consuming, but you can rescue a population with few individuals. When we talk about outbred population, it's much more complicated. So what you have heard is a huge experiment, I think, unique in the world. And when Marta says that she only doubled the populations at some point, this is crazy, okay? So Marta has the quality, lots of qualities, obviously, and one very important quality is that she's not only very joyful and very, energetic and enthusiastic, as you could see, but she is also able to, she's very organized, okay? And she can deal with several dozen students, which you might say, okay, so they did it all. It's a nightmare to have students with the type of work that she has been doing, where mixing populations is a disaster, losing populations of such a small size is crazy, and she didn't lose one, okay? So this is remarkable. And so she took a little while to deliver the thesis, and you can understand why. She was really burnt out. I mean, she was crazy. This was really crazy time. And she can do anything after what she did. And so I'm very pleased that she's with us again. So that's basically it.
Okay, thank you very much, and we can feel the enthusiasm of Marta Santos while she was talking about her work, and she infects us in a good way with her enthusiasm about this research. We now have some questions in the live chat, and so I will start with the questions by Patricia Beldade. I see two questions here on the live chat. So I will say the first two questions by Patricia. She says, I was a bit confused about the inter-replicate differences. I thought you said there was reduced consistency between replicates in fecundity for the lines evolved as large populations relative to small populations. So this is one doubt, and I think Patricia will add in the comments very briefly because she mentioned this was part 2 of 3. So Marta, I don't know if you already have some comment about this question by Patricia. We have already here the final comment by Patricia. I'm sorry, I was not seeing. But then when you showed the plots for actual variance, I thought you showed more variance for small populations. How is it? Thank you.
Okay, let me. I'm gonna share my screen again. Maybe it's easier. If I show the graphs, I think I know what Patricia is talking about the differentiation of populations. That was one of the puzzling results that we had. I guess this is the slide. So first of all, in terms of the direct response, in terms of starvation resistance, there was bigger response in large lines, smaller response in smaller lines, as expected. Then the problem was on the indirect response to directional selection, the fecundity. Here, I think where she was talking about here. So we did see a big decline in fecundity in the smaller lines, but in the larger, and if that happens, we think that it's the added contribution of the trade-off between starvation resistant and fecundity. So if there were more starvation resistant, they already had a lower fecundity, and then you add in breathing depression, and the fecundity is even lower. In the SGB lines, if we consider that the trade-off exists, so if there's bigger response to selection, then the drop in fecundity should be even bigger, and that didn't happen. And what we got was a huge difference between lines. So because there was no consistency, the average didn't low as much as expected. Then, when we talk about differentiation, and that was exactly what we saw. There was a reduction in variance among the small lines, and then in the large populations, we saw a high and a significant increase in variation. That was already found by Cohen in the 80s, where the combined effect of uniform selection and drift can have bring higher levels of variance. I think that was it.
Okay, thank you. Thank you, Marta. We have another question by Vitor Souza. So his question, he starts by thanking you for the interesting talk, and he has a question about the hybridization results. Was that a single pulse of admixture or continuous generations with gene flow?
Okay, I knew that question would be coming. So this was a massive hybridization. I'm sorry, it's a little further. This was a massive hybridization event. First of all, because we couldn't do the sequential hybridization. That happened once, and we mixed males and females from the populations we were hybridizing. There were 5 at the small lines, 2 at the larger lines per BI, and we did it all at the same time. Actually, Mark Phillips, my other scientific sibling, is doing some really cool work on yeast, showing that sequential hybridization is the only way of keeping or rescuing all the variation of the isogenic lines he's using. And here, that would be something we would like to do. But because we had to resume selection and pretty quickly, because reverse evolution would be in charge too if we didn't do it, we only did a massive hybridization scenario, and I'm pretty sure that influenced greatly the results.
Okay, thank you, Marta. So you already knew that this question was about to pop up. So I am checking here our live chat. For now, we don't have more questions, but you have more researchers from the center giving you congratulations about the nice work that you have done. So congratulations once again, Marta. And so if we don't have more questions, I know we have a slight delay from the transmission from the stream here from the Zoom to the YouTube. But if we don't have any more questions, and if the Professor Margarida Matos doesn't have further comments about the research, I would say that it's time for us to wrap up the session. And if further questions arise, you can always contact Marta Santos, right? My homonymous in the center. So Professor Margarida Matos, do you have any final remark to add?
I have a provocative question. Which is, if you were able to do a longer term experiment with these populations, what would you do? It's speculative. What would you expect? Because the small lines were slower responding, but you never reach a plateau. So do you think that they would catch up, taking longer time, but catch up with the large lines, or they would like stabilize at a lower value, which may be also very relevant, right, for conservation issues?
Yeah, it is. But if I need, we need to get that genomic data figured out. We need to do the experiments on the genetic data, on the genomic data of the large lines, because if we think about the classic expectations, they would never get there because they would lose so much variation. They would probably couldn't respond so well, and they would probably enter the extinction vortex, and I would lose the lines. But these new genomic data that say that they don't lose that much genome-wide variation. So maybe they just needed time. And because this form of selection depends always on the flies, because you just only select until they can handle it. We're not doing a super strong that can kill them in one generation. No, they if they are given time, I believe they would probably respond much more.
It's really an open issue, right? We would expect by traditional population genetics and evolutionary genetics that they would be at a lower value, because if the rate is slower, it would mean that maybe they are losing some variability, because otherwise the rate should be the same. But at the same time, there is inbreeding depression. So there are many issues. I think long term always tells something more, right? Basically,
Now, if you ask me, would it be worth it? No. No, well, not for me.
I understand.
I'll leave that to the next researcher, the next naive PhD student who would like to go on that journey.
Thank you. Thank you, Marta. And thank you, Professor Margarida Matos. You have a few more colleagues congratulating you. Luis Marques, for example, congratulating you for your great work. Also, Gabriela Rodrigues. And Patricia Beldad says she has a provocation, but she will talk to you separately afterwards about with a further question. And so I would say we can wrap up the session, the presentation now. I would thank again Marta Santos for accepting our invitation to be invited speaker in this CNT meeting. I thank Professor Margarida Matos for her presence here and for presenting Marta Santos and for all her comments. I thank Mariana Marques, who is on the backstage here, ensuring that everything goes well. And thank you to everyone that followed this presentation, this stream. And please stay tuned for the next CNT meetings and other events of C3C.